One of the first steps in basically all model systems in genomics was to try to produce a reference genome. And so that happened a little over 10 years ago within maize, and it was this line B73, which was developed by Iowa State, and Pat Schnabel, that's here at Iowa State, led the sequencing of that genome. And so for the last 10 or so years, our whole understanding basically of the genome of maize has been filtered through this lens of this single reference genome. And maize is an incredibly diverse species. So if you look across different lines of maize, there's genes that are present in some lines and absent in others. There's all sorts of structural variation within the genome where you've got sequences of DNA that are inverted or missing. So there's a lot more variation that's there than is present within that single line B73. So what we wanted to do is to get a much more broad and comprehensive survey of the diversity that exists within the species. So we worked to assemble this set of 26 lines that have been used for a long time in maize to represent the diversity. So it's basically a diversity panel. And it spans tropical maize and temperate maize and popcorn and sweet corn and field corn, all the main varieties of corn that are out there. So it's a real nice cross sample of the species. So a lot of the lines that we've sequenced are tropical lines. And so as maize moves, say, from tropical to temperate regions, what we found in doing this study is that particular genes that are important for disease resistance were lost during that migration from tropical to temperate. So these new genomes that we've assembled have some of these disease resistance genes in them that are missing within that B73 reference, which is a temperate line. So now we can capture those sequences and better understand where they're at in the genome, how they function, and potentially, through modern methods like gene editing, introduce those into temperate lines. So I hope we can move away from this single reference point of view, where we're funneling our understanding of the diversity of the genome through, just one line. I mean, it's a great line developed here at ISU, but there's so much more to maize than can be captured within that assembly. And now that we have these diverse sequences of maize, we really need to move forward towards better methods to really capture and harness that diversity and leverage it towards breeding. Up until now, we've had kind of a fuzzy view of what's important in terms of determining the traits or the phenotypes of maize. So the technology over the last couple of years has improved to the point where we're able to get these larger sequences. And now it's feasible to generate de novo sequences of maize at a pretty reasonable cost.